Welcome to the Deep Dive, the place where we take the most comprehensive, complex material in science and genetics and turn it into a streamlined, easily digestible conversation.
And today, we are undertaking a monumental journey.
We really are. We're tracing the evolution of our understanding of inheritance, moving from these abstract, invisible concepts all the way to the quantifiable chemical reality of the human blueprint.
Our focus today is the genome. And this isn't just about single genes anymore. It's about the collective, complete body of genetic information, the entire instruction manual housed within a single haploid set of chromosomes.
Right.
Our mission is to unpack its structure, understand its surprising complexity, and reveal what all these complete sequencing efforts are teaching us about human uniqueness and our deep evolutionary history.
The timeline of discovery here is genuinely fascinating because it's marked by these long periods of, well, quiet, followed by these explosive conceptual leaps.
Oh, absolutely. When you look at the historical milestones, the whole idea of inheritance starts conceptually in 1865 with Gregor Mendel's description of discrete factors.
But then it's this huge gap.
A huge gap. The truly transformative period hits in the mid-20th century. We move from the conceptual to the physical with the defining experiments between, say, 1944 and 1952, which proved definitively that DNA, not protein, was the genetic material.
And that all culminated in the pivotal year of 1953.
Yes, when James Watson and Francis Crick described the elegant double helical structure of DNA.
That structural breakthrough, which we'll definitely detail later, essentially launched the modern mission. Now, with the complete nucleotide sequences of species from bacteria to the human and chimpanzee genomes in hand, we can directly compare them.
We can map every duplication, every loss, and reconstruct the evolutionary pathways of life.
It's kind of the ultimate forensic tool for biology, isn't it?
It is. We can identify exactly which sections of the genome have been subjected to rapid change, what geneticists call positive natural selection, versus those that have just sort of drifted neutrally over time.
Okay, so let's unpack this enormous topic, beginning right at the foundation late in the 19th century with Mendel's quiet work.
We absolutely must start with Gregor Mendel in the 1860s. He was working on pea plants in Brno, focusing on crossing experiments, and his genius lay not just in the crosses themselves, but in his methodology.
Why?
He selected 7 specific traits, things like plant height, seed color, or pod shape, that were really clearly defined and only existed in 2 alternate forms.
And crucially, he didn't just observe, he counted. He counted the results of thousands of crosses. This quantitative approach was, it was unheard of at the time in biology.
It was revolutionary. And his conclusions, if we translate them into modern terminology, they basically define the field. He established that traits are governed by distinct particulate factors, which, we now call genes.
Okay.
Every organism carries 2 copies of each gene, receiving 1 from each parent. Those alternate forms, like the one that leads to a tall plant or a short plant, are known as alleles.
And he also noted the principle of dominance, where one allele, the dominant one, could completely mask the expression of the other, the recessive one.
Exactly. And building on that, he proposed 2 fundamental laws. The first is the law of segregation.
Okay.
This states that even though the pair of alleles for a given trait remain together throughout the organism's life, they have to separate or segregate during the formation of gametes, sperm, or egg cells.
So each reproductive cell only receives 1 copy?
And the second law is the law of independent assortment, which is, I think, maybe the more complex principle.
What did that one propose?
Independent assortment proposed that the segregation of alleles for one trait, say plant height, had absolutely no effect on the segregation of alleles for another trait, like seed color.
So they behave like independent variables during inheritance. It's like flipping 2 different coins.
Precisely. Now, this assumes, of course, that those genes are located on different chromosomes, a crucial distinction that would only be clarified much, much later.
What's so remarkable about this historical context is that his 1866 paper was a flop.
A complete flop. It was completely ignored by the scientific community, sitting dormant for 35 years until 1900, when 3 different European botanists, Hugo de Vries, Karl Correns, and Erich von Schermach, independently did similar experiments, got the same conclusions, and then stumbled across Mendel's original work.
The sheer intellectual tragedy of such a profound discovery going unappreciated for decades is just staggering.
It certainly is. But the pendulum was already swinging from the abstract to the physical. By the 1880s, biologists were looking inside the cell nucleus, watching cell division. They realized that while the liquid contents of the cytoplasm divided kind of randomly, the nuclear material divided with exquisite precision and equality.
And that precise, visible nuclear material, those threads that absorb dye, they were first observed by Walter Fleming and named chromosomes, which literally means colored bodies.
But the key question remained, are all chromosomes functionally the same? That answer came from Theodore Bover's really elegant experiments on sea urchin eggs. He studied polyspermy.
That's the fertilization of an egg by two sperm.
Right. Exactly. Which naturally results in disaster. Boveri found that these polyspermic eggs led to chaotic cell divisions and the death of the embryo early in development.
So what did he deduce from that?
He correctly deduced that the extra sperm contributed an extra set of chromosomes and an extra centriole, disrupting the whole orderly process. His conclusion was profound. Orderly development doesn't just require a set of chromosomes, but a specific particular combination.
So a qualitative difference among the chromosomes. Not all chromosomes carry the same information.
Exactly. Not all are equally necessary. At the same time, we had the realization that the chromosome number has to remain stable across generations.
Right.
Eduard van Beneden observed in the roundworm askers that the gametes, the pronuclei, had exactly half the number of chromosomes compared to the adult somatic cells.
And this led August Weismann to propose, even before the physical process was observed, the necessary existence of a reduction division,
which we now call meiosis. This division halves the chromosome number to prevent the count from doubling with every single fertilization.
And then the physical evidence that really tied Mendel's abstract factors to these physical structures arrives with Walter Sutton in 1903.
Yes. Working with grasshopper tests, specifically the spermatogonia, he counted 23 chromosomes, and he noted they existed as 11 look-alike homologous pairs plus an unpaired accessory chromosome.
And he showed that the behavior of these homologous pairs during meiosis was the perfect physical analog for Mendel's law of segregation.
It was a perfect match. They existed in pairs, they stayed together during the organism's life, and then they separated, with each gamete getting just one copy. This gave us the chromosome theory of inheritance.
But Sutton himself identified the huge potential flaw in this theory almost immediately.
He did. He realized if genes are physically arranged on chromosomes like beads on a string, then all the genes on one chromosome should be inherited together as a package.
They should form a linkage group.
A linkage group. Yet, Mendel's 7 traits, upon which the entire foundation rested, all assorted independently. This was the big conceptual challenge they faced.
So it was a serious conflict that needed resolution.
It was, and that brings us to the next great phase of genetic investigation.
So moving into part 2, the resolution of that linkage problem relied heavily on shifting research to a new organism, the fruit fly, Drosophila melanogaster.
Drosophila was an ideal model organism for 3 main reasons. A short generation time of only 10 days, the ability to produce hundreds of eggs per mating, and the ease and low cost of maintenance. This allowed for really rapid, large-scale breeding experiments.
Thomas Hunt Morgan started his work in 1909, but he needed variation, right? He spent a lot of time breeding thousands of wild-type flies until he finally found his first mutant in 1910.
And it was a fly with white eyes instead of the normal brick red. This was a pivotal moment. By systematically crossing and analyzing this white-eyed fly and other subsequent inheritable variations, Morgan and his students had identified 85 distinct characteristics by 1915.
And this body of work provided strong evidence for spontaneous inheritable changes in genes mutations as the source of all variation.
It established a critical link to evolutionary theory. And when they analyzed how these 85 mutations were inherited, Sutton's prediction about linkage groups was immediately confirmed.
They fell into 4 distinct linkage groups,
which correlated perfectly with Drosophila's 4 pairs of homologous chromosomes. Genes were, in fact, linked.
However, the linkage was incomplete.
Right. Alleles that started together on the same chromosome would occasionally separate during meiosis. Morgan proposed the mechanism in 1911, inspired by earlier cytological observations by F.A. Janssens, who had seen homologous chromosomes physically wrapped around each other during prophase of meiosis,
forming those visible X-shaped structures called chiasmata.
Yes. And this wrapping and exchange was the process of crossing over, or genetic recombination. It provides the physical mechanism for genetic variation, allowing alleles inherited from the mother and father on separate homologous chromosomes to exchange pieces and be reshuffled onto a single chromosome.
This is where Alfred Sturtevant, who was just an undergraduate in Morgan's lab, had his critical flash of insight.
It's an incredible story. He proposed that the frequency of this recombination could be used as a proxy to measure the physical distance between any two genes or loci on a chromosome.
Walk us through the genius of that logic. It's so simple and elegant.
It's purely physical geometry. If two genes are very close together on the chromosome, there's a very low probability that a crossover event will happen to occur precisely in that small space between them.
So they'll almost always be inherited together.
Exactly. But if the two genes are far apart, there's a much greater stretch of DNA where a crossover can occur, leading to a much higher recombination frequency.
So measuring the recombination rate provides a numerical unit of distance. A 1% recombination frequency means those genes are one map unit apart.
And this allowed Sturtevant to construct the first detailed linear maps showing the precise serial order of genes along the Drosophila chromosomes.
The ability to manipulate the rate of variation followed with H.J. Muller's work in 1927.
Yes, he showed that exposing flies to X-rays drastically increased the rate of mutation. This provided a powerful tool for accelerating genetic research, but it also served as an early warning about the mutagenic hazards of radiation exposure.
And the physical validation of Sturtevant's abstract numerical maps came from a stunning visualization technique, the rediscovery of giant polytene chromosomes.
In the salivary glands of Drosophila larvae by Theophilus Painter, these structures are incredible. They're about 100 times thicker than normal chromosomes because the cells undergo repeated DNA replication, sometimes up to 10 rounds, creating 124 side-by-side DNA strands without ever dividing.
And when you stain them, they display about 5,000 distinct, repeatable bands.
The critical finding here was that specific mutations could be physically located and correlated with individual bands on these giant chromosomes. The relative physical distances between the bands matched the relative genetic distances calculated purely from Sturtevant's recombination frequencies.
So it was a visual confirmation of the accuracy of the genetic map.
A direct visual confirmation. And what's more, these polytene chromosomes aren't just static roadmaps. They showed dynamic regions that swell or puff out at specific developmental times.
The chromosome puffs.
And these were identified as sites of extremely active DNA transcription, offering a direct visual demonstration of which genes were being expressed at any given moment in the organism's development.
So, okay, we know the genes exist. We know where they are physically located on chromosomes, and we even have maps and pictures of them in action. But what are they made of?
And that takes us into part 3, the critical shift to the chemical identity of the genetic material. The consensus for a long time, unfortunately, favored protein.
And the logic was simple, right? Proteins are made of 20 different amino acids, which allowed for immense complexity.
Exactly. DNA, based on the prevalent but very wrong tetranucleotide theory of the time, was thought to be this monotonous, simple repeat of ATGC, which just seemed incapable of encoding the immense variety of life.
But the seeds of the truth were already there. Let's just quickly look at the basic chemistry. DNA is a polymer made of repeating units called nucleotides.
And each nucleotide is built of 3 components: the 5-carbon sugar deoxyribose, a phosphate group at the 5th position of the sugar, and one of 4 nitrogenous bases at the 1 position.
And those bases are classified by their structure. We have the single-ring pyrimidines, thymine or T and cytosine or C, and the double-ring purines, guanine, G, and adenine, A.
These nucleotides link up to form the linear DNA strand. The sugar and phosphate groups link together via 3'5" phosphodiester bonds, creating this robust, repetitive sugar-phosphate backbone, while the bases project inward.
And that linkage gives the strand polarity or directionality, which is so crucial. It always runs from the free 5', which carries the phosphate group, to the free 3' end with the hydroxyl group on the sugar.
This chemical understanding was refined by Erwin Chargaff in 1950. He systematically analyzed the base composition of DNA from many different species, and he shattered that monotonous tetranucleotide theory.
His findings were twofold. First, the base ratios varied widely between species, which proved complexity.
And second, he found this profound numerical relationship that held true for every single species. The amount of purines always equaled the amount of pyrimidines. Specifically, the amount of adenine always equaled thymine, and guanine always equaled cytosine.
A and G equals C. Chargaff's rules.
Chargaff's rules. This provided the critical chemical constraint that was necessary for understanding the physical structure. It was the vital clue that led James Watson and Francis Crick, leveraging data from Maurice Wilkins and Rosalind Franklin's X-ray diffraction images, to propose the double helix structure in 1953.
This is where the synthesis of chemistry, math, and crystallography just hit a spectacular high point.
Right.
The Watson-Crick model laid out 12 key elements that define the structure. Let's not list all of them, but what are the absolute highlights?
Okay, the most critical points are these. First, DNA is 2 chains of nucleotides, not 3. Second, they spiral around each other to form a right-handed helix. Third, and this is absolutely essential for function, the 2 chains are anti-parallel.
Meaning, if one strand runs 5 to 3 fit, the other must run 3 to 5 fit, like 2 highways running in opposite directions.
A perfect analogy. Fourth, the sugar phosphate backbone is on the outside, and the bases project inward, stacked like a pile of plates.
And they're held together by hydrogen bonds between those paired bases.
Right. Weak individually, but tremendously strong collectively. The width of the molecule is maintained at a constant 2 nanometers because base pairing is always between one pyrimidine and one purine.
The single ring and the double ring.
Which leads to the most important point. The pairing is highly specific, and this solves Chargaff's rules. A pairs only with T, forming 2 hydrogen bonds, and G pairs only with C, forming 3 hydrogen bonds.
And that fixed geometry allows for unlimited sequence variety while keeping the structure completely stable.
Yes, and finally, because of this, the 2 chains are chemically complementary. The sequence of one strand absolutely dictates the sequence of the other.
That complementarity is the revelation. It's what ties the structure directly to function, specifically storage and replication.
Exactly. The immense sequence variety satisfies the requirement for storage of genetic information, and the complementarity immediately suggested the solution for replication and inheritance.
This is the semi-conservative replication idea.
Correct. Watson and Crick proposed that the 2 strands separate like a zipper, and each exposed strand serves as a template to direct the assembly of a new, perfectly complementary partner strand.
Which means the only primary function left mysterious at that point was the third one, the expression of the genetic message. How the sequence dictates the assembly of proteins.
That would occupy molecular biologists for the next decade. But before that, there's the problem of packaging.
Right. The DNA in every single human cell is 6 feet long. It can't just exist as a relaxed floppy string.
It has to be compacted.
Yeah.
Jerome Vinograd's work in 1963 showed that circular DNA molecules, like those in viruses or mitochondria, are almost always twisted upon themselves. They become supercoiled.
And this supercoiling is not random. It's almost always negative supercoiling.
Which means the DNA is slightly underwound, having slightly fewer than 10 base pairs per helical turn. And that underwinding is key.
Why is that?
It creates torsional strain that actually helps in the critical task of strand separation. To replicate DNA or transcribe genes, you have to separate the 2 strands. The inherent negative supercoiling provides some of the energy to facilitate that separation.
But maintaining, relaxing, or creating supercoiling needs specialized enzymes, the topoisomerases, the molecular scissors and tape for DNA.
We distinguish between 2 main types. Type topoisomerases are the relaxers. They create a transient break in just one strand of the DNA. This allows the intact strand to pass through the gap, relaxing excess supercoiling that builds up. They are essential for preventing knots and strain.
And type 2 topoisomerases are for the much bigger jobs.
Yes. Type 2 enzymes create a transient break in both strands of the DNA. This lets them pass another full segment of DNA through the break before resealing it. Think about the staggering logistical nightmare of disentangling 2 replicated sister chromosomes, each 6 feet long.
They perform that crucial decatenation function.
Exactly, ensuring that the 2 daughter chromosomes can physically separate before cell division. And this role is so vital that it's exploited in cancer treatment.
How so?
Human topoisomerase II is the target of several powerful cancer drugs like etoposide and doxorubicin. These drugs bind to the enzyme, let it cut the DNA, but then they prevent it from resealing the break. In rapidly dividing cancer cells, this catastrophic failure to reseal DNA quickly leads to genomic destruction and cell death.
That is a phenomenal transition from fundamental chemistry to clinical application. Now, let's leverage this concept of the genome and move into part 4. How do we even measure the complexity of the genome itself?
We begin with the definition again. The genome is the collective genetic information in a single haploid set of chromosomes. To study its complexity, researchers first had to understand how DNA strands behave when they are separated and then allowed to reunite.
Which involves the process of DNA denaturation or melting by heating the DNA. The hydrogen bonds break and the strands separate.
And we can monitor this by measuring UV absorbance. When the strands separate, the bases are no longer stacked tightly, and that leads to an increase in UV absorption. The specific temperature where half the absorbance shift is completed is the melting temperature, or $2.
And it's a quick way to gauge DNA composition, right? Higher GC content means a higher $2.
Oh, exactly, because of those 3 hydrogen bonds. And the reverse process, renaturation, or reannealing, is the crucial analytical step. If you slowly cool the solution, complementary single strands find each other and reassociate to form a stable double helix again.
And this reannealing process gave us a tool to measure genome complexity.
Yes, using reannealing kinetics. When researchers compared simple genomes, like the T4 virus or E. coli, the renaturation followed a single symmetrical curve.
So what was the key insight from the speed of that reannealing?
The key insight, developed by Roy Britton and David Cohn, was that the rate of reannealing is inversely proportional to the complexity of the genome.
Okay, so a simple virus genome is like trying to find your friend in a small room fast. But a human genome is like trying to find them in a massive city.
That's a great analogy. In a small, simple genome, even if the total DNA concentration is the same, there are far more copies of each unique sequence. So complementary strands are highly concentrated and find their match very quickly.
But when Britton and Cohn looked at mammalian DNA, they got a shock.
A big shock. The DNA fragments reannealed at markedly different rates, showing these complex, jagged curves. This was the first experimental proof that eukaryotic DNA was organized into sequences present at wildly different concentrations. The curve showed 3 distinct kinetic steps, corresponding to 3 major classes of sequences.
Let's break those down, starting with the fastest fraction to reanneal, the highly repeated fraction.
This fraction is about 1 to 10% of the total DNA, and it zips back together instantly. It consists of very short sequences repeated over and over in large tandem clusters. We can categorize this into 3 main types.
Okay.
First, satellite DNAs, which are short, 5 to a few hundred base pairs, forming massive clusters, and they are typically localized in the structurally important centromeres. Second, mini-satellite DNAs, which are longer, 10 to 100 base pairs. These are highly polymorphic. Their length varies a lot from person to person, and this variation is the basis for DNA fingerprinting in forensics.
Got it. And the third?
Microsatellite DNAs, the shortest repeats, just 1 to 9 base pairs, scattered throughout the genome. They're used extensively to trace human population relationships because they have a high mutation rate.
And we can actually see where these are using fluorescence in situ hybridization, FISH.
We can. FSH uses fluorescently labeled DNA probes to physically locate specific sequences on the chromosomes. For example, using a probe for alpha satellite DNA, you can visually confirm that these highly repetitive sequences are concentrated exactly at the centromeres, confirming their structural role.
So next we have the middle tier, the moderately repeated fraction.
This is the most varied group, 20 to 80% of the genome, repeated from a few times up to tens of thousands of times. Some of these have known coding functions, like the genes for ribosomal RNAs and histones proteins we need in huge quantities.
But the vast majority of this fraction is non-coding.
Right. It's mostly dispersed, interspersed sequences grouped as signs and lines, which, as we'll discuss later, are the relics of jumping genes.
Finally, we reached the non-repeated single copy fraction.
This fraction reanneals the slowest of all because its complementary sequences are present at the lowest concentration, just 1 copy per haploid set. This is the fraction that carries the unique inheritable information, including the genes for almost all proteins.
And the truly massive conceptual surprise that came from the Human Genome Project relates directly to this final fraction.
It does. Less than 1.5% of the entire human genome is actually dedicated to encoding the amino acids of our proteins.
Just 1.5%. The rest is structure, regulation, or repetition. That completely reframes how we think about the organization of life.
Absolutely.
Before we delve into the evolutionary implications of that complexity, we have to address one of the most devastating manifestations of repetitive DNA, the trinucleotide repeat expansion diseases.
These are known as dynamic mutations because the repeat length is unstable, often expanding dramatically as the allele is passed from one generation to the next. They involve repeating units like CAG or CGG.
And the diseases fall into 2 distinct molecular categories. Type I diseases are strictly neurodegenerative.
Right, and they result from the expansion of the CAG trinucleotide within the coding region of the gene, and CAG codes for the amino acid glutamine.
The classic example is Huntington's disease.
Yes. If the normal HD gene exceeds about 35 CAG repeats, the resulting Huntington protein is synthesized with a polyglutamine track that makes it toxic. It's a dominant disorder, a classic gain-of-function mutation.
So the protein gains a harmful aggregated property.
Exactly. The toxicity comes from the fact that these aggregates sequester essential transcription factors like TBP and CBP, effectively starving neurons of the gene expression they need to survive. And this expansion also explains genetic anticipation,
Where the disease gets worse and starts earlier in successive generations.
because the repeat track tends to get longer when passed to offspring.
So type 2 diseases operate on a completely different principle.
A totally different principle. They involve expansion of various trinucleotides, often CGG or CTG, and they occur in the non-coding portions of the gene like the 5' non-coding region or an intron.
And the expansion is huge, right? Into the thousands of repeats.
Yes. The primary type 2 example is Fragile X syndrome, caused by CGG repeats in the 5' non-coding portion of the FMR1 gene.
And here, unlike Huntington's, we see a loss-of-function mutation.
Precisely. When the CGG repeat count soars past 200, the expanded allele is subjected to massive selective inactivation. The gene is effectively shut off. This prevents the gene from ever being transcribed or translated, leading to the resulting mental retardation and physical symptoms.
It is striking how the location of the repeat coding versus non-coding can flip the outcome from a toxic protein to a complete loss of the gene product.
It's a powerful illustration of genomic architecture.
Okay, shifting gears, let's move to part 6 and discuss the stability and purposeful instability of the genome over evolutionary time, starting with large-scale duplication.
We have to first recognize that in evolutionary history, entire genomes can duplicate. This is called polyploidization.
Resulting in offspring with, say, 4 homologous copies of every chromosome instead of 2.
Right. And while it's often lethal in animals, it's remarkably common in flowering plants like wheat, coffee, and bananas.
The conceptual importance here, first championed by Susumu Ono in 1971 with his influential 2R hypothesis, is that this provides a sudden, massive excess of extra genes.
Ono proposed that the evolution of complex vertebrates from simpler invertebrates was facilitated by 2 separate rounds of whole genome duplication in the ancestral lineage.
So 2 rounds of the whole genome just doubling.
Exactly. This essentially created redundant copies of every gene, allowing one copy to maintain its original necessary function, while the duplicate copy was free to acquire a completely new or specialized function over evolutionary time.
And what's the evidence for this hypothesis?
It comes from comparative genomics, particularly comparing vertebrate genomes to simpler chordates like Amphioxus. Vertebrate genomes typically contain about 4 times the number of certain genes when compared to the homologous sequences in Amphioxus,
Which strongly supports the idea of 2 duplication events.
followed by extensive gene loss and subsequent diversification.
So while whole genome duplication is rare, gene duplication of just a small chromosomal segment occurs much more frequently.
Yes, the mechanism for this is typically unequal crossing over during meiosis. If homologous chromosomes misalign, the exchanged material results in one chromatid gaining an extra segment, a duplication, and the other losing one.
Most of these duplicates are evolutionary dead ends, right? They become pseudogenes.
Most are lost or rendered non-functional, yes, but the few that survive can diverge and acquire new functions, leading to the creation of complex multi-gene families.
And the best example of this is the globin gene family.
Absolutely. Hemoglobin, which we use to carry oxygen, is a tetramer composed of 2 pairs of polypeptide subunits, the alpha type and the beta type.
The pathway of its evolution is a perfect illustration of this duplication and divergence.
It is. An ancestral gene probably had 4 exons and 3 introns. That consolidated into the modern globin gene with 3 exons and 2 introns. Then, around 500 million years ago, a critical duplication event created separate alpha-type and beta-type globin genes.
Which then separated onto different chromosomes.
Yes, chromosome 16 for alpha-globin and chromosome 11 for beta-globin in humans. And subsequent smaller duplications created specialized members within the family, embryonic forms, fetal forms, and multiple adult forms, each finely tuned for different stages of life.
And within these clusters, you find those non-functional vestiges, the pseudogenes, like sabetabelit.
Evolutionary relics. And it's estimated the human genome contains around 19,000 such pseudogenes. Researchers are only now discovering that many of them might actually be transcribed into non-coding regulatory RNAs.
So the genome is shaped by massive events like duplication. but it's also much more dynamically unstable than anyone previously imagined, thanks to transposition.
The concept of jumping genes was truly radical when Barbara McClintock first proposed it in the 1940s. She was observing strange, unstable mutations in maize, these unusual patterns on the corn kernels.
And she deduced that genetic elements were autonomously moving from one chromosomal location to another.
A process she called transposition. And like Mendel, her work was largely ignored for decades.
So they just ignored her for decades.
For decades. It wasn't until molecular biology confirmed the existence of these transposable elements, or transposons, in bacteria in the late 1960s that her insights were vindicated.
Wow.
Bacterial transposons generally use a cut-and-paste mechanism. They encode an enzyme called transposis that recognizes the ends of the element, snips it out, and inserts it into a new target site. This usually leaves a little telltale duplication, a footprint, at the insertion site.
And eukaryotic genomes are just saturated by these elements. At least 45% of our DNA is derived from transposons.
Right. And while most of these are old, non-functional relics, their mobility is still significant. Transposon insertion accounts for about 1 in 500 disease-causing mutations in humans today.
So eukaryotic transposons fall into two categories. You have the DNA transposons, which use that cut-and-paste mechanism.
But none of those are currently active in the human genome. They're all remnants of ancient activity. The ones that are currently active and causing genetic change are the retrotransposons, and they use a much different amplifying method, copy and paste.
Okay, explain that mechanism.
The element is first transcribed into an RNA intermediate. This RNA is then copied back into complementary DNA, or cDNA, by an enzyme called reverse transcriptase, which is encoded by the transposon itself.
And that new cDNA copy is then integrated into a new genomic location.
Meaning the original copy stays in place and a new copy is added. It's an amplification mechanism.
And the two major active families in humans are both retrotransposons.
That's right. First, the lines, long interspersed elements, specifically L1, which are over 6,000 base pairs long. We have 500,000 copies, and L1 mobility still contributes significantly to genetic variation between individuals.
And then the SIEs, short interspersed elements like the ubiquitous ALU sequences.
ALU sequences are only about 300 base pairs long and are found at over 1 million sites. Uniquely, they seem to have appeared relatively recently, about 60 million years ago, and they amplify by hijacking the reverse transcriptase encoded by L1 elements.
So these things are often called the genetic scrapyard, but they aren't purely destructive. They're raw material for evolution.
Absolutely. They can carry adjacent fragments of host DNA when they move, creating new composite genes. Their sequences are found in gene regulatory regions. Even more profoundly, functional genes have evolved from them. Telomerase is thought to be derived from an ancient reverse transcriptase, and the enzymes for antibody gene rearrangement from a transposase.
Our journey culminates in Part 8, using the tools of modern sequencing to read the complete genetic blueprint and interpret the footprints of biological evolution.
The Human Genome Project was an immense global effort, providing a rough draft in 2001 and a highly accurate sequence by 2004. However, the most profound discovery was the gene number shock.
Let's linger on that. Prior estimates were high, 50,000 to 150,000 protein-coding genes, but the final count settled around 21,000.
21,000?
Wait, 21,000, that's it. Roughly the same as a microscopic worm. How is that even possible?
That forced a fundamental reassessment of what defines biological complexity. Clearly, our complexity as humans is not simply a function of gen count.
So if it's not the sheer number of genes, how do we account for the massive biological difference?
The difference is in the utilization of those genes. There are 3 key mechanisms. First, alternative splicing. Over 90% of human genes may use this process, where different combinations of exons from a single gene are spliced together, allowing one gene to encode multiple distinct proteins.
It vastly increases our polypeptide repertoire.
It does. Second, non-coding RNAs. A huge fraction of the genome is transcribed into RNA molecules, like microRNAs, that don't code for proteins but instead regulate the expression of other genes.
And the number and complexity of these correlate with organism complexity.
Directly. Sponges have about 10, humans have over 1,000. And third, systems biology. Complexity emerges from the intricate, dense networks of proteins within the cell. A small increase in the complexity of these networks can lead to an exponential increase in organism complexity.
This realization underpins the principle of comparative genomics. If it's conserved, it must be important.
Exactly. Functional sequences coding, or regulatory, are subject to purifying selection and evolve slowly. Non-functional sequences evolve neutrally and rapidly diverge.
And when comparing the human and mouse genomes, which diverged 75 million years ago, we find large blocks of shared gene order.
But what's truly provocative is the level of sequence conservation. About 5% of DNA sequences are highly conserved between human and mouse. Since only about 2% accounts for known coding in regulatory regions, this leaves us with 3% of the genome that is highly conserved but entirely functionally mysterious.
The dark matter of the genome.
It is. It suggests significant unidentified functional roles. It's a huge area of research.
The ultimate comparative study is between us and our closest relative, the chimpanzee. We diverged 5 to 7 million years ago, and our genomes differ by about 4%.
To understand what makes Homo sapiens unique, researchers look for genes that show evidence of recent rapid change, those subjected to positive selection in the human lineage.
A prime example is FOXP2, a brain-specific transcription factor involved in language and speech.
The human FOXP2 protein differs from the chimp version by just 2 amino acids. Mutations in the human gene cause severe speech disorders. The human version regulates over 100 target genes differently than the chimp version, and these changes became fixed in the human lineage within the last 120,000 to 200,000 years.
Strongly suggesting a role in the evolution of complex human speech.
Another key rapidly evolving sequence is HAR1, Human Accelerated Region 1. It's not a protein-coding gene. It's an RNA encoding gene that has undergone an astonishingly rapid evolution in humans' 18 base substitutions, despite being highly conserved in other vertebrates. And it's expressed primarily in the developing fetal brain.
And for a simple yet powerful example of modern adaptation, there's the AMY1 gene.
This codes for salivary amylase, the starch-digesting enzyme. Chimpanzees have only one copy. Humans, particularly those in populations with a historical high-starch diet, show multiple copies, a form of copy number variation, CNV.
This arose via gene duplication and shows how rapidly genes can change under environmental pressure, even within the last few thousand years.
And this forensic analysis even extends to our relationship with ancient hominins. Initial sequencing of Neanderthal mitochondrial DNA suggested no interbreeding with modern humans.
But the nuclear genome told a different story.
Svante Pääbo's nuclear genome sequencing corrected that. While modern European and Asian DNA is 99.84% identical to Neanderthal DNA, critically, 1 to 4% of our nuclear DNA is derived from Neanderthals.
So interbreeding did occur after modern humans left Africa.
It did, with the contribution mostly concentrated in genes related to the immune system.
Moving inward to variation within our species, while we are 99.9% similar, 2 randomly selected human genomes still differ by about 3 million sites.
Sites that vary in at least 1% of the population are termed genetic polymorphisms. The most common type is the single nucleotide polymorphism, SNP.
A single base pair difference.
Then beyond single base changes, we have structural variation, large changes involving segments from hundreds of base pairs up to millions. A typical human carries about 1,000 such variants.
The largest of these, over 1 kilobatter, result in copy number variation, CNV.
Right. Duplications or deletions that affect 10 to 15% of the genome. As we saw with the AMY1 gene, CNVs are a significant source of functional variation and disease risk.
This wealth of sequencing data drives Part 9, the immense promise of genomic applications in medicine. Traditionally, genetics focused on high-penetrance Mendelian diseases like cystic fibrosis.
But the challenge now is tackling common, complex diseases like cancer, diabetes, heart failure, which are polygenic, influenced by multiple genes and environmental factors.
And the primary tool for finding those low penetrance factors is genome-wide association studies, GWASs.
So you're basically looking for tiny spelling differences, SNPs, that show up more often in people with the disease than without it. They don't identify the cause directly, but they point to the region of the genome involved in the risk.
Give us a landmark example of what GWA revealed.
A classic example is the identification of the ApoE4 allele as strongly associated with increased Alzheimer's risk. This SNP cluster, involved in cholesterol metabolism, helped link lipid processing directly to neurodegeneration.
But despite these successes, genes often run into the problem of missing heritability.
Yes. Geneses typically account for only a small fraction of the known inherited risk for complex diseases. This suggests the rest of the risk likely comes from thousands of very low penetrance variants, or perhaps rare, moderately penetrant variants that are missed by these studies.
Genomic data is also transforming therapeutics via pharmacogenomics.
This is using SNP data to predict an individual's drug response. For example, individuals homozygous for specific SNPs in the TPMT gene can't efficiently metabolize thiopurine drugs used for leukemia. Knowing their genotype allows doctors to adjust the dose safely and avoid life-threatening side effects.
To manage the millions of SNPs, geneticists leverage the fact that they are inherited in large blocks.
These blocks are called haplotypes, essentially giant multigenic alleles. A haplotype is defined by a small number of key tag SNPs. Once you know those, you know the identity of the entire inherited block.
And the International HapMap project was an enormous collaboration dedicated to mapping these common haplotypes.
Which drastically simplifies future association studies. A fascinating recent example is the haplotype associated with lactose tolerance. This ability is linked to a long haplotype that has undergone intense positive selective pressure in populations with a history of dairy farming.
So it's an example of rapid, recent human evolution driven by culture and diet.
Exactly, and with sequencing costs falling so rapidly, routine individual genome sequencing will become commonplace, allowing for far more direct and precise associations between individual variants and disease risk.
Finally, we turn to the dramatic part 10. The essential experimental pathways that definitively proved DNA was the chemical nature of the gene.
This story begins surprisingly early, in 1871, with Friedrich Miescher, who isolated a large, acidic, phosphorus-rich substance from white blood cell nuclei, which he termed nuclein.
The term nucleic acid came a bit later.
Yes, in 1889. But early researchers, seeing that chromosomal stains also stained nuclein, began to suspect its importance. But, as we mentioned, the idea was derailed by Phoebus Levene and the incorrect tetranucleotide theory. The scientific community just sided with proteins.
The first crack in that dam came from bacteriology, from Frederick Griffith's 1928 discovery of bacterial transformation.
In Streptococcus pneumonia, this bacteria exists in two forms: the virulent encapsulated S-type, which kills mice, and the non-virulent non-encapsulated R-type.
And Griffith's key experiment involved injecting mice with a mixture of two things.
He killed virulent S-type cells, plus live non-virulent R-type cells.
So individually, both should have been harmless. The S cells were dead, and the R cells were non-virulent.
Right, but the mouse died, and live virulent S-type cells were recovered. Griffith concluded that a potent, invisible transforming principle from the dead S cells had somehow genetically converted the live R cells into the specific virulent S-type.
Which proved that characteristics could be inherited via this transforming agent.
The Rockefeller Institute took up the chase, culminating in the 1944 paper by Oswald Avery, Colin MacLeod, and McLean McCarty. They aimed to isolate the chemical identity of that transforming principle.
How did they do it?
They meticulously purified the active substance from an extract of S-type cells, and their definitive evidence rested on chemical analysis and enzyme treatment. They showed it had the exact chemical properties of DNA, was free of detectable protein, and most crucially, only enzymes that specifically digested DNA DNases could inactivate its transformative capacity.
And enzymes that digested RNA or protein had no effect.
No effect. The evidence was logically conclusive, but the paper was met with skepticism because the belief in protein was so deeply entrenched.
The final, decisive experiment arrived in 1952 with Alfred Hershey and Martha Chase.
Using bacteriophage viruses composed solely of DNA and protein that infect bacteria. Their reasoning was elegantly simple. The genetic material must be the substance that enters the host cell and directs the assembly of new viruses.
So they set up two batches.
Batch 1 had the phage DNA labeled with radioactive PNPA. Batch 2 had the protein coats labeled with radioactive 3.
Phosphorus for DNA, sulfur for protein.
Got it. After allowing the phages to infect the bacteria, they used a Waring blender to shear off the remaining unattached phage coats outside the cells.
And the results were crystal clear.
When cells were infected with 3 labeled phages, the bulk of the radioactivity, the protein, remained outside the cell. But when infected with p-labeled phages, the bulk of the radioactivity, the DNA, entered the cell and was passed on to the viral progeny.
And that experiment, combined with Chargas rules and Avery's work, removed all remaining scientific obstacles. DNA was accepted as the sole genetic material,
paving the way for the immediate acceptance of the double helix structure a year later.
What a monumental journey. From Mendel's peas to the double helix and the complexity of the modern genome. Let's try to summarize the biggest essential takeaways from this deep dive.
First, the foundation. Genetic factors reside on chromosomes, and although genes are linked, that linkage is incomplete due to the physical exchange of material via crossing over.
And the physical carrier of that inheritance is DNA. an anti-parallel complementary double helix.
Second, the surprising complexity. Human complexity is not driven by gene quantity, that shocking SIM $21,000 gene count, but by functional enhancement mechanisms like alternative splicing and the regulatory power held by non-coding RNAs.
Third, genomic dynamics. The genome is shaped by massive instability, from ancient whole genome duplications, Ono's 2R hypothesis, to ongoing transposition via lines and signs, which drives both evolution and diseases like trinucleotide repeats.
And finally, application. Modern genomics, particularly G to B, is starting to reveal the polygenic, low-penetrance genetic risk factors for complex diseases, leveraging the existence of stable, inherited haplotypes to simplify our understanding of human variation and guide future personalized medicine.
And we leave you with our final thought, a reminder of the mysteries we still face.
If sequence conservation implies critical function, what essential, non-coding, non-regulatory roles are being played by that 3% of the genome that is highly conserved between humans and mice, yet completely uncharacterized? The quest to understand the genome's dark matter is only just beginning.
Thank you for joining us for this deep dive into the core concepts of the gene and the genome. We hope this has provided you with the clarity and detail you need to master this fascinating material. We'll catch you on the next deep dive.